In this video, we will cover PeakLab's model discovery option. You can see this feature as a kind of advanced model discovery for a single peak. PeakLab has 860 built-in peak models, and many of these are applicable to chromatographic peaks. In this example, we will use a single chromatographic peak from the early portion of a gradient run. By all appearances, this peak is still behaving as if it's part of an initial isocratic hold. In PeakLab's predecessor, there were really only two chromatographic models that could manage the nonlinear fronted and tailed shapes in a concentration series: the HVL or Harhart-van der Linde diffusion model, and the NLC, the Wade-Thomas nonlinear chromatography kinetic model. In PeakLab, there may be hundreds of models or more that outperform the HVL and NLC, some by a wide margin. To make it as easy as possible to understand which PeakLab models are most useful for a certain class of peak, we have this model discovery option. It works as follows. If you enter any one of the fitting procedures with data containing just a single peak, and the automated placement detects just one peak, there will be a right-click menu option, Search for Optimum Model Single Fitted Peak. This opens the automated model discovery procedure. In this particular case, we've checked the basic chromatographic functions and the HVL and Gen HVL models that have an IRF convolution in the model system. We simply click OK to initiate the automated fitting. One after another in sequence, the different peak models are fitted using reasonable starting estimates for the different parameters in each of the models. Unless the starting estimates are well removed from those appropriate to the data, or the model is itself not capable of fitting the specific shape of the peak, the fits will be very fast, as you see here. This option tests not only the core models, but the collection of built-in IRF or instrument response functions. In a short time, you will have a good sense for which of the many peak models in the program fare especially well in fitting this specific peak data. In a science as complex as chromatography, there may be many reasons why certain models perform better, and we have selected an example here that illustrates this point. At this stage, the fits are complete, and we have a summary of the models with goodness of fit and parameter values. In this case, we sort by F statistic, and we also report the statistical error in parts per million. A 1 part per million error is an R squared with 6 nines after the decimal point. The F statistic is probably the most widely used information criterion for determining the model that most effectively describes a given set of data. Unlike the ppm criterion, each additional parameter must make a significant contribution in order for the F statistic to increase. Here we see something interesting. The best F models for F statistic are twice generalized models, meaning that both third moment and fourth moment adjustable parameters are significant. This fourth moment term in a twice generalized model is typically needed only when directly fitting gradient peaks where the gradient has introduced a compression in the peak shape. For this example, the presumption of an isocratic shape was made as a result of the gradient baseline not being as yet apparent. In this case, however, the presumed isocratic peak shape is not true. Some measure of gradient is impacting the peak shape. When this compression is present, the fourth moment parameter compacts the fourth moment or kurtosis, the power of decay changing from a power of 2, a Gaussian decay, to a power that is higher than 2. Here we see that power being 2.07, 2.08. This model discovery simultaneously determines which of the instrument response functions, or IRFs, best describes the instrumental distortions in the peak shape. In this case, the best model is a twice generalized chromatographic model with just a single first-order kinetic component. This is a useful observation since peaks which elute in a gradient often have only a small exponential IRF remaining. This is because the gradient in the opposite direction cancels much of the IRF, leaving only a small measure of the exponential component remaining. In the model discovery procedure, the grade models are those where the fitted parameters failed statistical significance, the probability of a parameter being different from 0. This is useful for seeing models that may be overspecified with too many parameters, or instances where the fitting may not have realized the optimal iterative solution. We will now use this information to fit this specific peak. We will first replicate the data 3 additional times in order to do a model experiment. Right-click replicate this data set a specific number of times. We will pick 3. We will go into the local maxima peak option. We'll select model experiment, and we're going to pick the 2 closed form solutions, one with just a third moment, one with the third and fourth moment. We will pick the best model from the model discovery, and we're also going to include the model I'm showing you here. That is a deconvolution model. It's a model that directly fits the gradient as the reverse of an IRF, that which unwinds the sharpened peak as if an isocratic separation had occurred. Now the full fit procedure is more elaborate, consisting of many discrete fits that enter into the overall fit, and it's much more likely to converge to the optimal solution for a given model. These fits are very fast, less than a second. We will go out now and we will look at these 4 fits, all of which look quite good. The best are the last 2 that you see here. The model with the IRF has the same that we saw in the model discovery, a 1.74 part per million error. The red is the deconvolution of the white peak, which is the observed, showing the difference between the peak with and without the instrument response distortions. And you can see the IRF is still quite significant when you're fitting in this manner. In the case of the deconvolution model, where you're unwinding the gradient peak, you see the estimated isocratic peak is very different from the gradient peak. Both cases confirming that the initial peak was not an isocratic peak. It was being impacted by the gradient in the mobile phase portion of the program.